welcome to the Novogene Introduction to RNA-seq Analysis, Content and Pipeline webinar. This is the second in our lockdown webinar series. We hope you will join us for the rest of the series. We'll be publishing details on that later. Bit of housekeeping. The webinar will last an hour and there'll be a 10-minute Q&A session at the end. Please submit your questions via the Q&A function. You'll see it at the bottom in the middle of your screen. We'll answer as many of the questions as we can live or direct in the functionality there. If not, we'll get back to you via email. I'd like to introduce your host for today. This is Gherkin Korkmaz. Gherkin did his studies in biotechnology in Germany and Sweden. His research focus at Uppsala University covered bacterial protein synthesis and codon usage bias. After his academic path, he moved to Cambridge as an account manager at Illumina and joined Novogene in April 2019. Since the beginning of the month, he's been promoted to lead the DAC team at Novogene. Congratulations, Gherkin, and over to you.
Thank you very much, John, for the kind introduction. Also from me, a very warm welcome to our webinar today. My name is Gherkin Korkmaz. As John said, I'm the sales manager for the territory covering Germany, Austria, and Switzerland. A little bit before we start the webinar, I'd like to introduce you to the company. Novogene just recently celebrated their 9th anniversary since its founding in March 2011. So we're quite a young company still. Our mission from day one was and still is to provide cutting-edge genomic technologies and high-performance computing to our customers to improve life, science research, and human healthcare. So Novogene has one of the largest sequencing capacities in the world. The cumulative output of all our instruments is at around 280 whole human genomes per year. So this is not only supported by our NGS instruments in our laboratories, but also by our supercomputing data centers that we have on site. Novogene was founded 2011 in Beijing. So our headquarters is still there. We do have subsidiaries across the world. To name a few, we have subsidiaries in California, Singapore, as well as Cambridge, where we have our lab for the European market. And most recently, we have opened a subsidiary in the Netherlands, and that will serve our customers better in the European Union. So if you are a postdoc today in a lab in, let's say, Germany, and you finally receive your well-deserved tenure position in Spain, we can arrange a smooth transition for you, okay? So you don't have to worry about one thing less. This slide answers the question, what kind of sequencing services we provide? The short answer would be, we do basically everything that NGS has to offer. In the early days, we were very active in the de novo genome sequencing field, where we have participated in quite a few prestigious de novo projects. Of course, we're also involved in resequencing plant and animal, as well as human whole genome sequencing, as well as whole exomes. For human WGS, we not only serve our customers from the research area, but also for the diagnostics. Of course, let us not forget our microbial genomes. Here we can support you as well for de novo projects, as well as resequencing, amplicon, or microbiome profiling. If you are interested in genetic regulation, we can support you in the epigenetics field, namely WGBS, bisulfite sequencing, ChIP-seq, as well as RIB-seq, if that is something you can benefit from. So transcriptomics will be our focus today. What we can do here, we have several different library options for you. So poly-A selection, stranded, non-stranded, ribosomal depletion, or iso-seq can help you learn more about alternative splicing events. as you can see, there are really not many limitations on what sort of assay we can provide to you. Even though we are a global company, we are also very well positioned on the local level. We do understand that sometimes it is just better to have a meeting in person on site. So for this, we have currently 21 local representatives throughout Europe, and they are more than happy to discuss with you your projects more in detail, and if you prefer, also in your local language. So the easiest way to contact us is through our email, europe@novogene.com. So once you write an email to us here, we will distribute your question to the responsible account manager, and he or she will then take it from there and discuss in detail with you how we can support you in your NGS needs. So that's already enough from me. I would like to introduce you now to our speaker for today, our Senior Product Manager, Dr. Wei Li. Wei received her master's and her PhD from the Clemson University in the United States. She worked on the cotton transcriptome. After her academic career, she started in the bioinformatics department as a senior bioinformatics engineer here at Novogene. And very soon after that, she was promoted as our current senior product manager. She has been giving seminars across Europe last year. I think it was 5 countries, every day a different university on campus. It was quite successful. So she's a great source of knowledge and a great educator, and I'm sure all of you will be appreciating her talk today. So as John mentioned throughout the talk, you'll have the opportunity to ask questions, and then we will try to answer as many of them after Wei's talk. Wei, after to you.
Okay, thank you for the introduction. Now I'm going to share my screen. So thank you again to join us for this webinar. And today I will give you an introduction about the RNA-seq analysis contents and pipelines provided by Novogene. And in this webinar, we will cover 3 portions. First, we will give you an overview about the major RNA-seq service that we are providing right now. And then we will dive deep into the eukaryotic messenger RNA-seq analysis pipeline. And then I will introduce you some specific difference in other RNA-seq service. So first, in the eukaryotic messenger RNA-seq or non-coding RNA-seq portion, as shown on the top, you may see different type of RNAs may either compete or interact with each other. And to explore each type of the RNA, we will have different services by corresponding to each tab. And for the messenger RNA-seq, we can provide sequencing from NGS platform or PacBio platform. But for the non-coding RNA-seq, we usually will run it on the NGS platform. And we also provide the whole transcriptome sequencing to explore the network between multiple types of RNAs. To analyze those data, if you are running a messenger RNA-seq, we don't need a reference genome available because we already have a very well-developed de novo pipeline ready for you. But for other non-coding RNA-seq analysis, we do need a reference genome to run the future steps. For the prokaryotes, if you are interested in a single species, then you can perform the very standard prokaryotic RNA-seq to check the transcript quantification and their structure information. If you are interested in the microbial community or multiple species in the community, then we can offer you the meta-transcriptome to study which species is the most or very active in the micro community. Well, for each project, we will run through this workflow. from sample preparation, we check the sample quality, we constructed the library. We also accept pre-made libraries if you have your special library. And we will check the library quality as well. Then we proceed to the sequencing and the analysis. For the projects without bioinformatic analysis request, we will deliver raw data and the data QC report to you after the project is complete. And for the project with bioinformatic analysis request, we will deliver raw data, analysis report, and publication-ready analysis results to you. Then let's go into the second part to see how we can perform the analysis in Novogene. Usually for the RNA-seq analysis or projects, we care two questions the most. So first, what they are, like what kind of transcript isoform has been expressed in my samples. And the second one will be quantification, like how many times of my gene has been transcribed into those isoforms. So basically for majority of the RNA-seq analysis, we will address both questions in our pipelines. And usually when we got the data, we will map them back to the reference genome and then perform the annotation to let you know what they are and the quantification to let you know how many there are. Here I am showing the messenger RNA-seq pipeline in details to let you know how we can perform it. So every project will start from the raw data on the top, and then we clean up the data, and we only use the clean data for the future steps, including the mapping or alignment, and then gene expression quantification and the associated analysis, and also the structure analysis, including those three different sections. And for the sections squared in the orange box, so those will be only applied for some human and mouse projects for the disease research. So everything starts from the raw data, and we really want to make sure our raw data are good for use. So for the data produced by the NGS platform, all of them will be saved in the FASTQ format. And in each read, you will see 4 lines for each read. And the first and third line are showing the sequence identifiers and descriptions. And the second line is the real raw data or the real sequence that we're going to use. So it will include ACTG or N for uncertainty. And the fourth line will be the corresponding quality for each base in the second line. So in another word, the length of the second line will be always equal to the length of the fourth line. And from the quality summary, we can calculate or we can check the distribution of our data, the quality distribution of our data. So E will stand for the sequence error rate, and the Q-FRET score is -10 log 10 E value. So you can see the relationship between those two symbols. They are negatively correlated with each other. And in our graph, showing on the bottom, so left and the right is showing read 1 and read 2 respectively. So as the reads elongate, we will see increased or accumulated error rates along the position. As we can see from this equation or the relationship, we will expecting lower and lower quality along the position as showing on the left. So this is how we can check our data quality by using the quality data from our sequencer. And in Novogene, we usually guarantee unsurpassed data quality for a project. So in some projects, we can guarantee Q30 higher than 80%. What does that mean? It will show you more than 80% of your total bases will have maximally error rate at as 1 out of 1,000, or minimum correct rate as 99.9%. So this quality guarantee actually exists the Illumina official guarantee. And the second step, we will check the base distribution along the race. So as Y is showing the percentage of the bases, so we are not expecting a huge separation between AT curves or GC curves as shown on the right. So it's kind of a good distribution for us to move forward. And the third one will be the data filtering. So in this step, we will classify all your raw rays into different categories. If the rays has any adapter sequence, that will be adapter-related rays. If the rays has so many uncertainties, more than 10%, that will be the containing N rays. And if the rays are with low quality, like more than half of them have very low Q-score, that will be the low quality rays. And the last portion will be the clean rays. So for the project without bioinformatic analysis, we will release raw data together with the pie chart as a reference for you. But for the project with bioinformatic analysis, we will, before we move forward to the analysis, we will remove all those bad rays and only use the clean data for the future steps. So here we got our clean data ready, and then we move forward to the next step, which will be the mapping. So in this step, we'll map all of our rays, all of our clean rays to our reference. And from this step, we want to know where they mapped to and if this region they got a map belongs to any gene. So to get those information, we will need two files from our reference genome. One will be the FASTA file. In this file, you can check your chromosome information and also the actual sequence of each chromosome. And the second file will be the annotation file. So from this one, you will check the gene information and their position, their gene ID, their gene name, and also the annotation source. So with those information, we will tell if we got any read mapped to any region of my genome, we can tell if this region belongs to any gene. So this step will give us those kind of information by alignment. And in your, before we move to the analysis, we will provide you a list for the reference. So you can search your species keywords like the common name or Latin name to check if we have any reference available already in Novogene. And all those information are collected from those three popular genome website browser. And if you have any in-house genome you want to use, or you cannot find the perfect version you prefer to use, you can always fill in our BI form with the genome information and any download information for us to access to your preferred genome version. So through mapping, you will get a lot of information, and we save all of our results in two different formats. First, we save all the numbers or counts in a table as shown on the top. And we also present all your data in some sort of popular format of the figures. Like for the mapping, we will use this pie chart to show you which genome region your race mapped to. Like we got exome region, intron region, or intergenic region. And also for this figure on the right, you can also see the mapping status distribution along each chromosome. And this is a human project, so you will see all the 22 plus X and Y chromosome distribution in the figure. And we can also visualize the data by using our outputs. So you have to use the IGV software, and in this software, you can load your reference files. We will also provide those files to you in the data release package. And you also need to load our mapping results, which will be saved in the BAM format. And with that, you can select a specific region, zoom in, and check the sequencing coverage or sequencing depth for each position. Through mapping, then we will be able to quantify our rays. However, we don't use our raw counts to perform the comparison to call our differentially expressed genes directly. Because if we use the raw rays, and once we got any different length for the gene or different gene size, such as if we have the gene B, which is much longer than gene A, it's quite possible that we got more mapped rays on gene B versus gene A in the same sample. And also, if we got different total read counts output, like we got 10 total rays for the sample A, but 10 million rays for sample B. So it's also will affect our rays quantification. Like we may got more rays in the red genes in sample 2 versus in sample 1. So that's why we usually normalize them before we perform any further comparison or visualization. So FPKM is a common way to normalize our RNA-seq data. As its name indicates, it is fragments per kilobase per million. That means we normalize our fragments counts for each gene against the two factors. One will be gene length, and the other will be the total read counts. So through this normalization, then we can move forward for the next step. So after normalization, we will summarize the FPKM distribution for each gene and in each sample in a table. And we also demonstrate those tables in different type of figures. Like you can use the density curve to show the FPKM distribution, or you can use the box plot or violin plot. So all of those different formats are provided or included in your result package. With the FPKM output, we can also perform another two analyses. One will be the correlation analysis showing on the left and PCA clustering showing on the right. So both analyses will allow us to check if we got any good replications within my groups, or if we got a very nice treatment effect between groups. Such as on the left figure, if the color is getting darker or we have higher value, that means we have a higher similarity between each pair of the samples. While on the right, if we can cluster our samples or cluster our replicates within the group along either axis or principal components, then we may see a very nice treatment effect between groups. So from this one, we can use for those two purposes to check our replicates and our treatment effect. And another option that will be to pick up our outliers. So if you see any sample is responding very weird, so maybe it will be a very nice place to pick up our outliers. And then we will move on with the differential expression analysis. This is the most important step for the RNA-seq analysis, as we really want to call out which gene has been differentially expressed in my sample statistically. So in this step, we will not use FPKM to perform the analysis. We will only use raw read counts as input, as this is requested by the software. So the software used in this step will be either DSeq2 or EdgeR. Those two software both request raw data or raw counts as the input, because they have their own normalization algorithm in the software. So in this step, we need your information from the BI form. So you need to define which one you want to name as the treatment group and which one name as the control group. And through the differential expression analysis, we will call out the DEGs, differentially expressed genes. And in the table, we will show all details like the gene ID, their information in sample, in group, or their statistic information, and their annotation informations. And to present those data, we have different type of figures again. So you can either use bar graph to show the gene counts in different comparison groups, or you can also check the unique differentially expressed genes or common common differentially expressed genes among comparison groups. So that will be the Venn diagram. And the next two figures I'm going to show you will be the most popular figures in the RNA-seq result presentation. So one will be the volcano plot and the other will be heat map in the next slide. So volcano plot, in this one, we will plot all genes along the axis. So X is showing the log-to-fold change between the defined groups. Here, we will just use A versus B as a demo. And Y-axis is showing the negative log10 p-adjusted value. And here, dash line, this dash line is showing the cutoff where we define significance or not. So from this figure, if we got positive log-to-fold change, that will be upregulation in the comparison groups A versus B, and negative value will show the downregulations. And any dots above the dash line or the cutoff will be significant differentially expressed genes. So from this figure, we can tell we got around 4,600 significantly upregulated genes shows in red, and 3,300 significantly downregulated genes shows in green. So this can give you an overview about your data. And the next one will be the heat map. So in this heat map, we will cluster all the differentially expressed genes based on their expression profiles across different samples. So each column is showing different sample, and each line, actually we got so many DEGs, so each line is showing one differentially expressed gene. And red and blue shows up and down regulations respectively in this figure. And we will also label out their group sources as showing here, or their gene length, their gene tab, and their chromosome location. So if you check here, majority of them will be protein coding genes as indicated in the legend. So heat map is a very commonly used map for us to perform some functional prediction. Like if you have some gene that you have no idea what they are and what their potential biological functions in your sample, if you see any well-studied gene clustered together with your interested gene, then you may have a reference like my unknown gene may functionally similar as the well-annotated gene because they cluster together. That means they respond similarly in the same sample under the same treatment in my experiment. So because they behave similarly and we cluster them together, so that will give you a hint about their potential functions. And with the DEG list, we can move on with the enrichment analysis. So for differentially expressed genes, we will use different databases to run the enrichment analysis for different purposes, as shown on the top. And for the expressed genes, we will use gene set enrichment analysis or GC analysis for the enrichment. So all of those will give us an idea about which biological pathway or which gene function has been significantly enriched in my defined comparison groups. And here I just show you a demo to demonstrate how our result look like. And on the top, we'll give you all the detailed information saved in table. And on the bottom, we usually present our enrichment or top enrichments either in bar graph or scatter plot. And bar graph usually will show the top 20 enrichments. And in the scatter plot, you can also check the circle size to see how many genes has been associated with this top enrichment. And for the CAG enrichment analysis, we will also present the significant enriched pathway in the result and label the DEGs related to this pathway in color. We will also perform PPI, protein-protein interaction, for the RNA-seq data. And in this one, we will use the string protein interaction database. And once we input the DEGs, and we can predict the potential interactions and load the data into the cytoscape. And in this one, you can transfer all your proteins or genes into bubble or node, as shown in the figure. And if there is any potential relationship between any two of them, you will see an edge or a connection between them. So here we are presenting the PPI, or the network between the potential, for the potential interactions for all DGs, only up-regulated genes or only down-regulated genes. And all of those will be something that we can get after the mapping and quantification. So in this branch, and then we will move forward to the second branch, which is the structure analysis. In this one, you can perform your alternative splicing. Alternative splicing is a major way to contribute to the diversity of our transcript isoforms in the eukaryotes. And we will focus on those 5 different types of the alternative splicing events. And the sashimi plot will present the difference of the different isoforms within different groups. As shown on the right, so bottom is showing the 2 different isoforms. One of them is keeping the middle extron, and on the top will show our mapping status. So if you see the if you see the peak in the middle, that means we got more rays mapped to this extron in the middle. And the arc is kind of showing the junction between those 2 rays, and the arc is proportional to the aligned read counts. So with those kind of sashimi plot, you will see where my junctions are and which type of the isoform is showing more in my samples. As from the top, we are expecting more extron keeping isoform in the orange samples versus the red samples. And another analysis will be the variant calling. So we will focus on 2 different types of variant. First will be the single nucleotide variant, which is SNP, and the second will be the multiple nucleotide variant, that is the indel, insertion or deletion. And those variants may cause different effects, like if they will change any amino acid or any codon in my protein level. So if there is no change, that will be silent variant. And if it is changed to the stop codon, that will be nonsense variant, or even missense variant if they change to other codons or other amino acids. And in our result, we will identify where they are. So where are my variant and their function, their potential function, like if it is a silent variant or it is a nonsense variant. And also we will predict their impact by using softwares to see how severe my variant it is to my sample. And the last analysis will be the fusion gene analysis. As you can see in the top example, so this is a research carried by the colon tumor. So in their research, they found a new fusion gene which come from the 2 separate genes. So those 2 will be the original 2 separate genes, and they identify a new fusion gene belongs to a small GDPase superfamily, and its expression level is extremely high if you compare to the other normal genes. So in a lot of disease research, fusion genes may be very important for like the tumor pathogenesis. So that's why we want to include it for some human and mouse study for the research, for the disease research. And we will also provide the results for you to visualize those fusion genes in the IGV. So you can load, again, the reference and your files in this software. And with that, you can check the potential junctions to see how my fusion genes has been transcribed from separate multiple genes. So in our data release, we will release report, result, SAS check files to see if you download everything, and BAM file, reference file for the IGV visualization, and also raw data and instruction for each part. And in the result package, you will see each folder, and each folder will correspond to the previous slides I've shared with you, and each folder will also go back to each step we have mentioned or emphasized in the previous slides. And we will also release a software list for you to check, including the software name, the version, and the parameter we have used in our analysis. So that will be some details for the eukaryotic messenger RNA-seq pipeline. And now let's move on to the last part to show you some special analysis in other RNA-seq services. So I've heard that there are some audience are interested in the single cell RNA-seq. So I will just talk about it in very briefly. So 10x Genomics is a very popular platform to construct the single cell library. And in this one, they include two important information, the 10x barcode and UMI, to label each transcript and each cell. So from this platform, you can just input a single cell suspension, but our output will be the digital gene expression profile for each transcript from each cell. So this is a very high throughput platform for us to perform single cell RNA-seq. But for the analysis, if you skip the middle portion I covered by the square, so actually the analysis pipeline is quite similar. We start from the raw data QC, alignment, quantification, differential expression analysis, and all the rest related analysis. But for the single cell RNA-seq, as we need to track each transcript and each cell, so we're going to produce a gene cell matrix in the quantification step. And we will carry out multiple steps to process those data because we have different purpose to run the single cell RNA-seq. Once we got this matrix, we will perform the cell clustering. As in our cell suspension input, there may be multiple types of cells, and we want to cluster them back to their original cell type, as shown here. So different cluster is labeled in different color. By comparing those clusters, we will call out the marker genes, as shown here in this violin plot. So those three genes will only have very high expression level in cluster 3, as shown in red circles. So our single cell RNA-seq will help us to predict some biomarkers, some new biomarkers for the specific cell type. And what's next? We can also use those information to perform the cell type identification. Like you don't know which cell type the cluster 3 belongs to, but we really found a very well-studied biomarker or marker gene. Then you can use that information to identify your cell type. Or we can perform some cell developmental progress study to track how our cell is developing through this timeline, or by using those different biomarkers for different steps. So we will not talk about too much details about this. And if you are interested, we may leave it for our potential future webinar. And for the other non-coding RNAs, in addition to annotation and quantification, we will also include the target prediction, as they may target different type of RNAs, as shown in this figure again, to regulate our gene expression. And in our whole transcriptome analysis, we will focus on the networks of those multiple types of RNAs. So here I'm showing a publication supported by Novogene, and it is focused on the long non-coding RNA study. So as we've seen in the figure. So those will be the heat map to show the differential express the link RNAs or messenger RNAs. And in the table, we also present the target, the link RNA targets, potential targets prediction list. And for the whole transcriptome RNA-seq, instead, in addition to the basic or the standard analysis for each type of those RNAs, we also focus on their interaction or potential competition. So that's why we involve the correlation analysis between two types of those RNAs or co-expression network study among three types of those RNAs. And here is another publication supported by Novogene. So in the bar graph, we show the gene counts for, we show the counts for different types of RNAs. And on the right, we are showing their potential networks by using the PPI or by using the network demonstration from the Cytoscape software as well. So circle, square, and triangle is showing different type of RNAs. And for the prokaryotes, again, single species that will prokaryotic RNA-seq, and the micro community that will be the metatranscriptome. Because in the prokaryotic prokaryotes, they have different mechanism to perform the gene transcription or gene translation. So that's why we focus on the, in addition to the basic steps from data QC, mapping, quantification, we also include the gene structure analysis, such as the operon analysis, UTR analysis. So those will be the additional part for the prokaryotic RNA-seq. And for the metatranscriptome, as we want to know which species is super active in my community, so in addition to those basic pipeline, we also include the taxonomic analysis to predict the species present in my community and also show you the abundance of them. To process all those data and to guarantee our turnaround time, we have to carry a very powerful computing system. So that will allow us to storage that a big amount of data and also to process a big amount of our projects. So with that, that will be everything I want to share with you today. And now I'm glad to take any questions that you are interested in.
All right, thank you so much, Wei, for this excellent talk. So we try to answer as many questions as possible. So let's start with the first question from Chaitali. For analysis, can we ask you run a pipeline of user choice if we ask, for example, D62 instead of cufflinks?
That is an option because for each step, not each step, but for majority step, if there is multiple popular softwares involved in this step, we will have some choice for our clients. Like you prefer to use D62, then you can choose D62. But if you prefer to use HR, we can go back to HR. So it's a possibility for you to choose your preferred software. And you may want to double check with our either TS or account manager for the options that we have for you.
All right. Kabad is asking, is it possible for you to provide RPKM and TPM as well?
I think for some of the analysis, we use TPM. If that's what you preferred, we can discuss with our PI team to see if we can provide RPKM or TPM in addition to our FPKM.
Irabonosi is asking, can you use the results from DSeq2 or HR to perform correlation and PCA?
That's possible too, because those are just different inputs to perform the clustering or the PCA analysis. For us, by default, we use the FPKM to check their similarity between samples or among samples. But if you prefer, we can have that additional clustering as well for you. But for anything in addition to our standard pipeline, there may be some additional charge for that special request. But for that it's possible, but I will double check for the team.
Thank you, Wei. Mohammed is asking what you mean by clean reads. If reads are not clean, will it require to perform the sequencing again?
So for clean reads, we will go back to here. So as some of the reads, they may have adapter sequence, or they may have too many ends, like for the pair end 150 sequencing strategy. Either read should have 150 base pair-ish, but if you got so many ends, like 100 out of 150 are ends, but with only 50 bases with clear nucleotide, then it doesn't make too much sense to use those containing N rays. So that's why we want to clean up our rays first to move forward, and that will guarantee or not to ensure we get more reliable analysis results. So for the clean rays, as I mentioned here, we will classify all of our raw rays into those different categories, and any rays falls in those categories, we will not use them, but we will only use the clean rays that has not falls in those categories for the future analysis.
Right, then we have the next question from Zen. How do you evaluate the mapping rate? If our mapping rate is only 60%, can this be used for analysis?
I think it really depends on the projects and also the reference genome, because the mapping rate they may affected by the sample status. Like if we got two degraded input samples, like the RNA is degrading into very small pieces, then it's very possible for us to get pretty low mapping rates. Or if we got a very poorly assembled reference genome, and then we may see a very low mapping rate as well. But to evaluate our mapping rate, we will check across all the samples. Like if you send us multiple samples, like 10 samples, and all of them get a very uniform mapping rate, then it may be not a very big problem for your project. But if you have like 8 samples got 90% for the mapping rate, but like 60% for the other 2 samples, then we will go ahead to check if those 2 samples have any problem or issues doing the whole pipeline. So as I mentioned, it really affected by many factors, and we really want to discuss if it will affect very severe for our future analysis case by case.
Then we have another question from Asif. He's asking, I have RNA-seq raw data. Can you analyze the data?
Oh, absolutely. So we offer BI analysis only as well. So if you don't perform the library construction or sequencing with us, we can also do that. But we need to confirm first. So you need to our local representative will contact you to collect more details, like the platform you use for the sequencing, like the data format, if it is good for us to move forward. So what I will suggest is you may want to send us one of your samples or one demo data for us to double check if everything looks good, and then we will give you the confirmation, like what kind of analysis we can do for you.
Then Kabat is asking, single-cell RNA-seq, is it limited to human? Is it possible in plant, protoplasts, or tissues?
So the issue is for human is quite mature for the human study, but for plants and other animals, it is possible. For this, first you need to have a reference genome available for us to perform the analysis. This is a master request. And the second is there may be some kind of technical obstacle for the plant and animal, because for some of the species, it's very hard for us to get a very high input cell suspensions. So high cell, I would say high cell numbers or enough cell numbers for us to construct the library. Because for the 10x platforms, they ask for very high cell activity and very high cell number input. But you may have some other options not using the 10x platform to construct your library. So there are other some platforms like the BD platform, so they can you can visualize your cell activity during the cell selection or cell sorting. So with that, maybe it is more suitable for species that you cannot culture the cell suspension very easily.
All right, the next question comes from Anna. She's asking, if you have no reference of the organism, can you work with its sequence?
If there is no reference for the messenger RNA-seq, it is possible. So we have the de novo pipeline ready, like in this one. So if you don't have the reference genome, we can use the short reads to assemble a reference transcriptome and pick up the unique gene from each cluster as a reference, then quantify your short reads. But for other non-coding RNAs, it's not quite possible for us to perform the de novo pipeline.
All right. There is a question from Barbara, quite technical. Are you developing results in R or Python and Linux?
So it's kind of a combination. So we use R, we use Python, we use Perl, we use Linux system, and our BI team kind of create a whole pipeline to connect all the steps together. And we are using multiple programming languages to make it very efficient for us to move forward.
Another question regarding clean reads from Shamer. Is it okay to have RNA-seq clean reads of about 90% with almost 10% adapter-related contamination?
It is quite possible. So as you can see in my figure, actually this is almost 99% for the clean reads ratio. So it really depends on the samples again. As for the adapter containing reads, this may be because we got very small insert size from our library construction. So it really depends on your sample status. If your sample passed our QC criteria, so it's quite possible you got very nice clean reads portion like this. But if we have very poor sample, like some sample maybe classified as failed sample from our side, so we may see a high ratio for the adapter containing reads.
All right, we have a couple of minutes for a few questions more. Next one comes from Ramona. Which values are used for generating the heat map?
For the values? For this one, we are using the normalized FPKM value. I'm sorry. For this one, we are kind of normalized first and then use the FPKM value to show the heat map. I think this is what we used here, but I can double check after this. to give you the confirmed answer.
All right, another question comes from Kelly. If our reference is not in your species list, what information should I provide?
So as I mentioned in the mapping step, so here we do need two files. First will be the FASTA file and the other will be the annotation file. So what we will recommend if your species is not in the list, you need to provide us the accurate or exact scientific name of your species. And if you cannot find it here, we may suggest you to search against those three popular browsers. So they may have the genome version for us to use, because for the same genome, they may assemble them they may optimize their assembling and annotation every year or every decade. So you need to give us the exact scientific name and the version of the genome you prefer to use. Or if you are also not familiar with how can I locate my reference genome version, you can provide us the scientific name only, and I will ask our BI team to check for you if we can find a good version for you, and we will confirm that with you as well.
All right, thank you, Wei. Thank you, all of you, for your time and your questions. If we haven't answered them online, we'll come back to you and answer them to you personally via email. We do hope you enjoyed the webinar and found it useful. We will email you a link to the webinar recording and the slide deck, along with a very short survey that we'd really appreciate you taking the time to complete. So the survey will help ensure we make future webinars even more relevant to you and your research goals. So in the meantime, please stay in touch. You can follow us on Twitter and LinkedIn, or you can get in touch via our website at en.novogene.com. So we look forward to seeing you all on the next webinar. Thank you very much.
Thank you again.